Hello, everyone, and welcome. My name is Rula Deeb. I'm a senior principal at Geosyntac Consultants in California and the coordinator of the SERDP and ESTCP webinar series. I have the pleasure of facilitating today's event. This webinar focuses on DoD-funded research efforts to understand the effects of multiple stressors on marine mammals and terrestrial species. First, Dr. Enrico Pirota from the University of St. Andrews will discuss research efforts to advance the understanding of the cumulative risk of multiple stressors, specifically on marine mammals. Second, Dr. Joshua Lawler from the University of Washington will talk about his work, which explores the nature, drivers, and impacts of interactions among multiple stressors affecting at-risk species. Each of these two presentations will be followed by a brief Q&A session, and depending on how we're doing on time, we will conclude the webinar with a longer Q&A session. These next two slides provide instructions on optimizing your webinar experience. If you have not done so already, please download Zoom at the link shown here and provide it to you in your webinar registration confirmation email. If you cannot download Zoom, you can view the slides using a compatible internet browser such as Firefox, IE, or Edge, and by creating a free Zoom account. If you're unable to view the slides or if the screen freezes, try keying in Control and F5 to perform a hard refresh of your browser. And in case of continued difficulties, please download a PDF of the slides from the webinar webpage. We're also dropping a link to these slides in the chat box. And call into the conference line provided to you in your webinar registration email. An alternative option to view today's proceeding is to go to SERDP and ESTCP YouTube channel, as we will be live streaming the webinar. Today's broadcast will be listen only. You can submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A periods to get your questions in. We do encourage you to get them in well in advance of those sessions. And we ask that when you submit your question, just please add your organization name at the end so we can identify it when relaying the questions. One more reminder, the questions should be submitted in the Q&A box and not the chat box. Please reserve the chat box for comments related to technical difficulties. At this point, I would like to introduce Kevin Hires, who serves as the SERDP and ESTCP Program Manager for the Resource Conservation and Resilience and Climate Resilience Program areas. In this capacity, Kevin is responsible for identifying strategic research needs to support conservation, climate adaptation, environmental security, ecosystem management, and wildland fire management on DoD installations. Kevin brings nearly 30 years of experience at the interface of natural resource management and science. His academic and management work have focused on fire ecology, conservation of threatened and endangered species, and the science support of prescribed fire modeling to meet ecosystem management objectives. In terms of educational background, he received his master's degree in conservation ecology from the University of Georgia. Kevin, please proceed.
Thank you, Rula. I'm happy to welcome everyone to today's SERDP and ESTCP webinar. The next few slides will provide a quick overview of SERDP and ESTCP, which together are the Department of Defense's investments in the environment and installation energy, science, and technology space. The programs report to the Deputy Assistant Secretary of Defense for Energy, Resilience, and Optimization, headquartered at the Pentagon. SERDP is the Strategic Environmental Research and Development Program established by Congress as a partnership between the Department of Defense, the Department of Energy, and the EPA. SERDP addresses high-priority environmental science and technology opportunities that focus on the top priority DoD requirements. SERDP funds both fundamental research as well as advanced technology development that ultimately impact real-world environmental management. ESTCP is the Environmental Security Technology Certification Program in which we demonstrate and validate innovative environmental and installation energy technologies. These investments capitalize on past investments under SERDP and other research programs, and ESTCP is designed to transition technologies into the field. Especially important in all ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. Advances in science and technology provide the DoD with the tools needed to effectively manage and restore its assets to protect troops, their families, the public, and the environment. SERDP and ESTCP investments improve installation energy resilience, environmental cleanup, UXO remediation, resource conservation, and the sustainment and maintenance of defense assets. SERDP and ESTCP fund, monitor, and actively guide relevant and applied research, development, testing, and evaluation projects for the DoD's top environmental and installation energy challenges. Our programs currently fund over 600 active projects. To maximize research impact, SERDP and ESTCP collaborate with top talent across sectors. In 2023, we funded projects with 77 academic institutions, 91 federal performers, and 52 industry partners. Our innovation and technology development inform DoD policy to ultimately improve environmental and energy management. Our webinar series highlights outstanding projects across all our main focus areas, and registration is open for webinars through the end of the year. You can find additional information about those upcoming webinars at this link. All past webinars are archived and can be accessed using this link. Please save the date for the 2024 SERDP Symposium, which will be held from December 3rd through the 6th in Washington, D.C. Symposium registration is currently open. I would like to also remind you that a copy of today's presentation session can be downloaded from our webinar webpage. We would appreciate it if you can please take a moment to complete the survey that will pop up on your screen at the end of this webcast. At this point, I'll turn it back over to Rula to introduce today's first speaker. Thank you for joining us, and I hope you enjoy the webinar.
Thank you, Kevin. Our first speaker today is Dr. Enrico Perrotta, who is a research fellow at the Center for Research into Ecological and Environmental Modeling at the University of St. Andrews in Scotland. Enrico's work focuses on the behavior, psychology, ecology, and life history of marine mammals and their interactions with human activities. Since 2020, Enrico has been involved in several projects on the effects of multiple stressors on marine mammals. These projects have been funded by SERDP, the Office of Naval Research, and the Bureau for Ocean Energy Management. He has authored over 60 peer-reviewed papers and delivered over 50 presentations and posters. In terms of educational background, he got his undergraduate degree in biological sciences from the University of Milan, a postgraduate degree in marine biology from the University of Pisa, a master's degree in marine mammal science from the University of St. Andrews, and a doctoral degree from the University of Aberdeen in Scotland. Enrico, we're delighted to have you speak today. Please get started with your presentation.
Thanks very much, Rula, and hello everyone. It's a great pleasure to be here today with you all, representing our projects on understanding the cumulative effects of multiple stressors on marine mammals. Just as an outline of my presentation, I'll start with some background and talk about the population consequences of multiple stressor projects that are called PCOMs from now on. I'll jump straight to the results of both the working group and the case studies, and then I'll conclude with some lessons learned and the benefits to the Department of Defense. As you all know, we're here. Wildlife populations are exposed to multiple stressors, and assessing the cumulative effects of these stressors is required under many jurisdictions. However, as highlighted in that report by the National Academies that you can see on the right from 2017, current available science is not adequate to meet these regulatory demands, both in terms of the methods that are available and the data, which is really the rationale underpinning the population consequences of multiple stressors project. The project comprises a working group. You can see the members listed there, and a series of case studies. So we have two data-rich case studies on North Atlantic right whales and bottlenose dolphins in Barataria Bay, Louisiana. And then we have a technology case study on epigenetics and an associated case study on northern elephant seals that is funded under separate SERDP award to the University of California, Santa Cruz. Let's start from the working group. This is a group of experts representing different disciplines, spanning from physiology to behavior, population biology, and statistics. And the role really is to advance the understanding of cumulative effects on multiple stressors on marine mammals, both from a conceptual perspective and in terms of analytical methods. And of course, throughout the project, the working group has been really important in guiding and supporting the work of the case studies. We have a series of outputs that were generated together with the case studies, but also some outputs that were working group specific. And I'll talk about some of the results in the next few slides, but I want to mention that we're also preparing a synthesis manuscript that will summarize the lessons we've learned throughout the project. So as we were thinking about the analytical approaches that have been used to study the effects of multiple stressors, we realized that they sit on a spectrum, which we call the assumption spectrum, between data-driven approaches and process-driven approaches. So on the left-hand side of the spectrum, we have approaches that rely on a lot of data and therefore can make few assumptions, but and as such, they incur a low risk of bias. However, as you can see, they tend to be noisy, they have low precision, and importantly, they have limited predictive power beyond the range of doses that are measured. So an example of one such approach could be a typical habitat suitability study, where you model your occurrence of a species as a function of the underlying stressor surfaces. At the other end of the spectrum, where we have less data, we need to rely on some information on the mechanisms underpinning the system. And so we need to make more assumptions, which of course incur the potential for bias if those assumptions are wrong. But in contrast, you can see that we are reducing empirical noise, we're getting higher precision and increased predictive power. And an example of these mechanistic approaches could be an agent-based model where you have these simulated animals moving in space, interacting with stressors over time. And where you sit on the spectrum depends on the data that you have available, but also the question that you're trying to answer for management. Another important output from the working group has been the development of the PCOMS framework. It's a conceptual framework that was initially proposed as part of the National Academy's report that I was mentioning earlier. And it links the exposure to stressors on the left to the population status on the right. So exposure to stressors is affected by other ecological drivers, such as the climate, and it results into some doses that are acquired by the animal. The animal can then respond either physiologically or behaviorally or both, or it can accumulate those doses in a stressor burden box. We then move on to this important box of health, which is a collection of indicators that represent the ability of the animal to survive and reproduce successfully. So these include, for example, energy reserves, endocrine status, immune status. And then all of this is happening at the individual level, represented by the large box in the framework. If we stack up all the individuals, we can then say something about the dynamics of the population. So essentially, as we move from left to right, you can see that the relevance for management increases because generally conservation and management are interested in managing populations rather than individuals. But of course, also the availability of data decreases as we move to the right. So we will need to rely more on those mechanistic approaches towards the right end of the assumption spectrum that I was showing you earlier. The other aspect we've been dealing with as part of the working group is the management of cumulative effects of stressors. And we've been arguing for moving away from a traditional way of looking at cumulative effects, where the proponent of an action is asked to assess the incremental effect of the proposed action on top of the existing status quo, and towards a cumulative risk perspective, where we're focusing on target population and the cumulative risk from the stressors that the population experiences. So this involves identifying what populations are at risk and defining some management goal, for example, a threshold of population growth rate that is deemed acceptable. We can then use conceptual model to link the activity to the stressors they generate to the population to identify chains of concerns and decide which of those chains require further modeling. We can then progress them to the PCOMS model that again links the stressor to the population status to answer this question: does the risk exceed the threshold set by management? And if the answer is no, then we can continue monitoring the indicators, the stressors, and their effect to make sure that the system stays in balance. But if the answer is yes, then we can use the PCOMS model to decide which combinations of stressors we can feasibly try to reduce to bring the population below that threshold. And sometimes in this process, we might identify unpredicted changes in the population that might lead us to revise the conceptual model and identify additional chains of concerns that we should consider. Moving on to the case studies, and I should say that the case study results are still in progress, so some of these results are preliminary. Our first case study was on North Atlantic right whales. This is a critically endangered species exposed to a wide range of stressors and with a long-term data set spanning the last 50 years or so. So the case study team has been developing new approaches to study the health of individuals in the population. So for example, photogrammetry from drones to look at body condition, or microbiome from blow samples, again, to link with health. The other problem with these animals is that they select their prey at very fine spatiotemporal scales, which makes it really hard to predict where they will be and where the prey patches are. So we've been developing new ways to sample and model the prey that tries to tackle this challenge. And you can see there a map on the right where we are predicting the zooplankton density in Cape Cod Bay as a function of some environmental covariates. And then we've also been looking at ways in which anthropogenic noise could interfere with right whales by looking at interactions between prey density, anthropogenic noise, and the formation of foraging group. And so in that plot on the right, you can see that we're finding indeed that for increasing noise level, the quality of the zooplankton patches that we find our groups in seems to be decreasing, suggesting that indeed noise might be interfering with the ability of the animals to find good foraging patches. And in collaboration with the working group, we've been developing a PCOMS model for this species, which models the underlying health of each individual in the population. So you can see at the bottom the estimated health trajectory for this one female, as informed by a series of data streams at the top. So this female underwent a few calving events, a few entanglement events, and then it's really that entanglement towards the end of her life that caused a drop in health that led to her death. So with this model, we can compare the effect of multiple stressors on health and also assess their combined effects with the ultimate goal of quantifying the consequences for vital rates, so survival and reproduction, which is our ultimate goal. We can look at this from a population perspective. So on the plot on the left, each gray line represents an individual in the population with its estimated health trajectory, while the blue line represents the average health of the population over time. On the right, we can see how by reconciling the effects of these stressors at the level of this common currency of health, we can now compare their effect size, and we can start to say something about the relative intensity of different stressors on the health of individuals. We're currently looking at the combined effects of these stressors and multiple scales. And we're finding some inconsistencies across different model formulations that suggest that we are lacking some important information to make conclusive statements about those combined effects. And so this is really important because it's highlighting critical data gaps for future research. Moving on to the second case study on bottlenose dolphins, here we took a different approach. We've been using controlled exposure experiments where we're exposing animals in this population to approaches, controlled approaches by vessels, and looking at the probability of responding to that vessel as a function of their health. And the health in this population is very heterogeneous because these animals have been exposed to the Deepwater Horizon oil spill. So some of the health of these individuals is really, really bad. And we have different combinations of response metrics and health metrics. And indeed, for some of those combinations, we're seeing that the probability of showing a response is changing as a function of their health. So for example, in that plot, you're seeing how animals with lung condition are showing a reduced probability of responding by diving to the oncoming vessel. We've also been running a cross-sectional analysis of scarring as a function of health, where we found that the probability of showing a scar from a vessel interaction was associated with some aspects of health, particularly adrenal insufficiency, but not with lung disease. And as a way to investigate the mechanism through which lung disease might affect the ability to respond to the oncoming vessel, we're looking at respiration rates as measured from these radio tags. And we are indeed finding that the time between surfacings is associated with lung disease. So in that plot, you can see that the red points and the red line are below the black points and line, suggesting that the time between surfacings is reduced for animals with lung disease. Moving on to the technology case study, this has been focusing on epigenetics. So epigenetic modifications are reversible modifications to the DNA that don't involve a change in the DNA sequence. And these are accumulated over time, for example, as the animal age, but they can also occur as the animal is exposed to stressors or disease, for example. And so the first step is to fit an age clock. And here we have been fitting an age clock to bottlenose dolphin samples, skin samples in particular, from a large number of sites that you can see represented in that plot. And you can see that our age clock is doing quite well at predicting the age of these individuals based on the methylation patterns up to an age of around 30, where we start to see that deviation from the diagonal. And the next step is to see whether this technological tool can be used to assess the health of individuals. And we're finding indeed that the epigenetic age of these animals is higher when they are exposed to chronic stressors, for example, entanglement or the oil spill. So basically the age, the epigenetic age is accelerated compared to the chronological age, suggesting that these animals have experienced some stressors to their health. And we're also finding that the residual of age, so the difference between the epigenetic age and the chronological age, is associated with health and survival probability. So on the plot on the right, you can see that for increasing residual, there is a decrease in health. This relationship is very noisy, but we were very excited to see that there was a signal. And finally, we have the northern elephant seal case study. Again, this is led by the University of California, Santa Cruz. Their research has been focusing on two strands. They've been looking at a historical bank of tissue samples with contaminant information and cortisol, but also historical information and long-term data sets on reproduction of individuals and their foraging trips. And they're finding these connections between the performance during a foraging trip and the energy that the animal gains, and in turn, its ability to reproduce. So in that plot, you can see that an animal doing very well in a foraging trip gains a lot of energy, is not stressed, and reproduces successfully. So these are the white dots. In contrast, the blue dots represent animals that did poorly during the foraging trip. They gain little energy. They came back very stressed, and they did not reproduce, even though they were pregnant at departure. And then finally, we have those red dots. These are animals that were not pregnant at departure, but you can see that they were very stressed. So even though they did very well during that foraging trip, they had already abandoned that reproductive attempt. So this is teaching us that for these animals that have a very timed and strict phenology and life history, there are very short windows in which we can impact their ability to successfully reproduce. And the team has been looking at these questions also from a molecular perspective, trying to understand the underpinning molecular mechanisms to these processes. The second research strand has been focusing on experimental exposures to the CTH, which is a precursor of cortisol, together with acoustic disturbance. And the effects of these experimental exposures have actually been quite transitory. And in contrast, they're finding quite strong year-to-year variation associated with the environment, suggesting that environmental variation in this population might actually be the strongest driver. In collaboration with the working group, we've been developing a PCOMS model for elephant seals, where we were able to develop a model for the probability of giving birth to a pup as a function of the female's body condition. In contrast, given the available data, we were not able to predict successfully the survival or the implantation of these animals. And again, we're finding variability in body condition metrics from year to year that suggests the importance of the environment in this system. So some lessons learned, starting from the data and the methods. We are finding that long-term observations and health data are really critical to inform these models. And particularly for cetaceans, we need new way to be able to collect health data remotely. And epigenetics represents a promising avenue for us to do that. The bottlenose dolphin case study is showing us that experimental approaches can be very useful in cases where we can control some of the exposure and contextual variables, but there remain, with this species, there remain field limitations and other contextual variables that we cannot control, which make the logistics of the field and the analysis of the data challenging. However, when we don't have data, we can rely on mechanistic information to help address the data gaps. So again, thinking about that assumption spectrum, we can move towards the right end of the spectrum towards process-driven methods to try and fill up some of our data gaps with our mechanistic knowledge. In general, we're finding that individual level analytical approaches are more effective, which must be informed by longitudinal data on individuals. And this is again because these species are long-lived. So really to understand the life history of an animal, we need to be able to get the longitudinal perspective analyzed using an individual level approach. The Rightwell case study has taught us that we also need to have information on the spatiotemporal distribution of the stressors, and this can be really challenging for a species that range thousands of kilometers, they live for multiple decades, but this is really important because understanding exposure rates to stressor is really the first step to be able to quantify the combined effects to those stressors. So in general, we have been moving away from these largely academic conversations about whether the interaction is additive, synergistic, or antagonistic, and towards trying to quantify the cumulative risk of multiple stressors to a species of interest, which is ultimately what management is interested in. We're finding that PCOMS approaches for threatened species are different compared to PCOMS approaches for non-threatened species. And that's because in threatened species, the signal of the effects of stressors tend to be stronger. And in non-threatened species, in contrast, even though the signal is weaker, we might have larger population sizes and a greater ability to manipulate the setting, for example, experimentally. We're finding that environmental variation is an important driver that must be considered in cumulative risk assessments because it's often one of the most important stressors that the animals will experience. And then as a general point, understanding the population consequences of multiple stressor is a multidisciplinary problem. And so we've really benefited from having this interdisciplinary working group guiding the case studies throughout the project. So in conclusion, PCOMS modeling can be used to identify combinations of stressors that can be reduced to meet regulatory requirements. Its implementation in practice will depend on the life history, data availability, and conservation status of the species under analysis. But throughout species and approaches, this idea of and concept of individual health is central to understanding PCOMS because these species are long-lived and monitoring effects on population dynamics directly is often going to be unfeasible and not very effective because by the time an effect is manifested at the population level, it might be too late to act from a management perspective. And again, I want to reinforce this point that environmental variation, particularly related to climate change, will be one of the most relevant drivers to marine mammal populations going forward. In terms of benefits to the DoD, we've been generating new theory, new measures, and new data to understand the effects of multiple stressors on marine mammals. And we believe that these are applicable to other terrestrial and freshwater systems, particularly where we have these long-lived species that require that longitudinal perspective and that require these modeling approaches because we can't measure feasibly changes in population status directly. This will hopefully inform environmental impact assessments, for example, by the DoD, for example, on Navy ranges, where sonar is used in the context of other stressors that are affecting marine mammal populations. And this will hopefully improve the capacity to manage wildlife, reduce uncertainty, and ideally reduce the need for precaution. The importance of this project has been recognized by the regulators. So we've been in constant communication with NOAA, with BOEM, and with the Marine Mammal Commission, who have been interested in hearing about our progress and our results throughout the course of the project. And I want to close with some links to some of the papers that I've been mentioning. And I want to thank you very much for listening. If you have any questions, please feel free to ask.
Great, thank you so much, Enrico. Just a reminder, if you have questions, please drop them into the Q&A box that you can see at the bottom of your screen. For now, we've received a number of questions for Enrico. The first one is from St. Mary's College of Maryland. How did you determine the threshold for risk? Is it based on population size or different factors of the population?
Sorry, I couldn't hear. The threshold for?
Risk.
For risk. Yeah, that's a decision that it's in the hands of the regulator to some extent. So we think that those thresholds could be set in various ways, based, for example, on the dynamics of the population, the size of the population, the type of the species. But yeah, I mean, those thresholds are set by the regulator by and large.
Great, thank you. For the right whale case study, you mentioned that the combined effects appear inconsistent. Can you elaborate on that, please?
Yeah, so we're finding that some of the stressors are indeed combining to generate a stronger effect than when they're on their own. And I should say that because of model formulation, this is partly inbuilt into the model. But on top of that, we're also finding that, for example, the effect of carrying fishing gear from an entanglement is stronger when the prey conditions are poorer. However, we're finding that model formulation has some influence on the results. And so depending on model structure and assumptions, the conclusions on the combined effects of the stresses change to some extent. So these suggest that some of those assumptions are critical, and they seem to revolve around this question of where the animals are and how they get exposed to stressors. And so it's really highlighting, as I was saying earlier, those data gaps that should be targeted first. And an important step is, again, coming up with these surfaces for stressor distribution that can be fed to these models.
Great, thank you. This next question has to do with the bottlenose dolphin case study. What next steps do you anticipate taking for that study?
Yeah, thank you. So we're currently finalizing the interpretation of the results of those differential responses given the health of individuals. So we've been generating these dose response functions that link the probability of exposure, the probability of responding to the dose. And ultimately, the probability of responding relates to the risk of getting struck given your health. So we're considering ways to develop a PCONS model for this case study, where we will use those results to simulate alternative scenarios of stressors, for example, different scenarios of traffic or oil spill. And there is an existing population model for these populations, so it will allow us to simulate population trajectories. There are, of course, a lot of uncertainties when doing this, but we think it'd be a valuable demonstration to show how the experimental results could be used in practice to potentially inform management.
Thank you. This next question is from CDM Smith. Has exposure to contaminants been incorporated as a potential stressor encountered by marine mammals in your model?
Yeah, so in the case of the bottlenose dolphin in Barataria Bay, we are essentially we are essentially including the exposure to contaminants by using the individual health. So health acts as an aggregator, an accumulator of these responses or exposure to stressors over time. So these animals have been exposed to the oil spill, their health has been affected, and we're using this health as our in our model. In the case of the right whales, we haven't, and the reason for that is that even though it's a really important data stream, and we believe that it probably has some effect on the animals, we have a small sample size, and so it's given the size of the population, given the length of the time series, we don't have enough of these data points to be able to incorporate it in the model in a successful way. But this is something that we definitely are keeping in mind, and that we suggest should be addressed using future data collection, because we believe it's an important stressor.
Wonderful. And then again, specific to the model you described, what type of data do you need to collect or use to inform the health box in the framework, in the model framework?
Yeah. So this is a really important box in our framework. And as I was saying, it's made up of a series of indicators that represent really different physiological systems. So in the gold standard to inform that box would be blood analysis to measure various biomarkers, which in general requires some ability to handle the animals, which is of course very tricky for my mammals. So we have this problem of accessibility, and we also need to collect health data for a large proportion of the population. So this is possible for some small odontocete species. So it's routinely done, for example, for bottlenose dolphins along the east coast of the States, and for pinnipeds, but not for most free-ranging cetaceans. So we need new methods to collect this data remotely, and some have been explored in this project. For example, the microbiome sample from the blow, photogrammetry data to look at body condition from the drone, or epigenetics that can be sampled from skin samples, again, remotely. But there are others. So, for example, fecal sampling, blubber sampling, and there's current work on developing a tag that could sample the blood of animals remotely. So there is exciting development in the field, and the alternative, if one cannot measure health directly, is again to revert to mechanistic modeling. And this has been done, for example, for the energy part of health, where we've been developing bioenergetic models that describe the way in which animals maintain their energy budget.
Thank you so much. And one last question before we transition to the second and last presentation. Are the models that you are developing easily accessible by folks in the audience, and can they be adapted to other contexts and systems?
Yeah, so all the model code and the data, in fact, are made publicly available when we publish the papers. And this would be either statistical model to estimate effects from the data, or a simulation model that can be used to make predictions. And we believe that the structure and the principles that underpin this analysis are applicable to other systems and other contexts. And in fact, this is something that we want to cover in that synthesis manuscript I was referring to. So we're planning to discuss the applicability beyond the case studies that we analyzed. It's likely to be more relevant in systems that involve long-lived species, where measuring the effects directly on the population is unfeasible. But there are many of those, so we believe that this whole approach and the methods will be applicable, and yes, they will be available.
Wonderful. Well, thank you for a very engaging presentation. Please stick around. We'll try to answer any unanswered questions towards the end of the webinar, depending on time. But we're going to go ahead and transition to our second speaker, who is Dr. Josh Lawler. Josh is the Oren and Althea Sost Professor in the School of Environmental and Forest Sciences, the Associate Director of Nature and Health, and the Faculty Director of the Botanic Gardens, all at the University of Washington. Josh is a landscape ecologist and conservation biologist focusing on applied conservation questions and their real-world applications, with much of his efforts focusing on how anthropogenic factors affect species distributions, animal population dynamics, and community composition at regional and continental scales. Josh has published 136 peer-reviewed papers and book chapters. He served as the lead author on the 3rd U.S. National Climate Assessment. In terms of educational background, he received a bachelor's degree from Bowdoin College and master's and doctoral degrees in ecology from Utah State University. Josh, please go ahead.
Thank you, Rula. I'm going to talk today about our SERDP. First of all, thank you for having me. I'm delighted to be able to talk about our SERDP project, which focuses on multiple interacting stressors in terrestrial systems and using scenario-based modeling to explore those. I'd like to first acknowledge two of our co-leads on this project, Julie Heinrichs, who's with the Computation Ecology Group, and Gary Slater, who leads Eco Studies, the Eco Studies Institute. Slide 43, please, I think. All right, so I'm gonna just give you a little outline of the talk. I'll start with some objectives, and then I'll go through our approach, and then I'll talk about some initial results. And I've got those listed as initial results because we're a little more than halfway through our project, and I'll show you some of the outputs we have, but there'll be a lot more from this project still to come. And then I'll end with some relatively quick conclusions and some potential benefits to the Department of Defense. So our objective in this study is to build models and develop scenarios for exploring the nature, the drivers, and the impact of interactions among multiple stressors affecting at-risk populations. And the study focuses on 5 specific species on 2 different military installations. So we're working on the streaked-horned lark and the Taylor's checker spot butterfly and the Mazama pocket gopher on Joint Base Lewis McChord in Washington. And we're working on the black-capped vireo and the golden-cheeked warbler on Fort Cavazos in Texas. At the heart of the approach to this model, there's actually 2 key elements to the approach that we're using to explore interacting stressors for these 5 species. And one of those key components is the building of spatially explicit individual-based population models for each of those 5 species. And so these are agent-based models that track individuals through space and time. And at the heart of those models is information about demography, so reproductive rates, survival rates, demographic rates in general, and then also information about behavior. And at its very most basic, and what's included in all of our models, is movement behavior, and that can be dispersal behavior or other aspects of movement. But these models can also incorporate other behaviors such as mate finding or avoidance of stressors or other behaviors, interactions with predators, interactions with competitors. And then also involved in this model is the environment and how the environment impacts demography and behavior. And so as inputs to the model, not only are there parameters, demographic parameters and movement behaviors that are programmed in, but we also take spatial inputs, and those spatial inputs can represent habitat. They can represent the distribution of a stressor, and that could, you can imagine that could be a toxin in the environment, or it could be a predator or some kind of parasite. We also have spatial layers that represent barriers to movement or represent ease of movement across the landscape. And so these spatial layers are brought in, and they can be static, so they can be a layer that lasts throughout an entire simulation, or we can bring in layers that change through time. And so for all of our models, we have habitat habitats that change through time as driven by one or more or different stressors or different scenarios, which I'll talk about in a little bit. So as the model runs, each individual is tracked, and an individual will move, and then it might reproduce. Well, it might find a range, a home range, or establish a territory. It might reproduce with some probability, and then we have a survival stage where there's a probability of mortality, and then there's movement again, and that cycle goes around again. And that's a fairly simple version, but we go through that cycle for each individual. And then what comes out of the model is what you'd expect from a population model. We can take the outputs and we can plot population size over time in response to whatever scenario we've driven through that model. But we can also, because this is a spatially explicit model with the spatial inputs, and we're tracking the individuals through space, we can also plot where those individuals are through time and get an idea of how the distribution of the population, how densities of individuals in different areas has changed through time. To look at things like whether there are sources and sinks in the population, or whether the distribution is shrinking or growing or shifting in general. The second part of the approach, I mentioned there were two major parts of the approach, and the second part of the approach involves developing stressor scenarios. And so for the streak-torn lark, I've just demonstrated that there are several stressors, but there are three of them up here on this slide. And each of those stressors, we're developing different scenarios of how that stressor might affect the streak-torn lark. And so the image on the top is the Scotch broom, which is a major invasive species on joint-based Lewis McCord in the prairies, and it affects the habitat of the streak-torn lark and its ability to nest. And so we're developing scenarios that have to do with how quickly Scotch broom spreads and grows, which can be affected by climate change and other environmental factors, but also how Scotch broom is managed and how frequently it's burned or how frequently it's mechanically removed. And so we can build different scenarios that look at different management regimes and different spread of that invasive species in particular. Likewise, training exercises can have an impact on the lark, and we can build scenarios that have to do with when that training occurs, how frequently, and where it occurs, and what type of training occurs where. And then finally, fire is important in this system, as well as at Fort Cavazos. And so all of our models will have fire scenarios. And in this case, some of this is fire, wildland fire that's all generally started by humans, but some of it is controlled burns and some of it is not. In the case of the lark, as the climate changes, it may become more difficult to do controlled burns for managing habitat. And so we'll have at least one scenario that looks at how the ability to control burns as a management strategy for habitat may change through time. Slide 48. I'm now going to give you some initial results for each of the species, for each of four of the species. I'm going to talk about four of the species. And each time I show you some results, I'm going to start with a slide that looks like this. And this is a depiction of what the model for this species looks like and what it includes. On the right, you see a circle, and that's the life cycle that we've developed for the Mazama pocket gopher. You can see it includes dispersal, territory selection, reproduction, survival, and then there's a gray box for habitat change. The orange boxes are the ones that contribute to the results that I'm about to show you after I show you this slide. And the boxes to the left with the pointy arrows pointing into the circle represent either management scenarios or stressor scenarios. And some of those combine and are not totally independent. And the one that's in orange, extreme weather, is the one I'm going to talk about next. And there are several ways that extreme weather events can affect the Mazama pocket gopher negatively. Increased precipitation, so flooding and precipitation, heat events, extreme heat events, and extreme cold events can all affect the Mazama pocket gopher in its burrows. And so they, depending on when they happen and how frequently they happen, they can have differential effects on the population, particularly on young, if they're happening at a particular time of year. And so the simulations that we've run some simulations to explore an extreme weather scenario in which, as climate changes, extreme weather events become more frequent. Slide 49. The red line at the top of this slide shows what the baseline population does when we run simulations for that. So you can see it's a slow decline. There's actually variability. That line doesn't show it. But then all those colored lines represent the manifestation of an extreme weather scenario in which extreme weather events become more frequent, occurring in 1 out of 10 years on average. Each of the colored line represents a different run of the model. And on the x-axis, you've got the simulation year, and on the y-axis, you have the population size. And then you can see that those lines jump around. You can see that each of those lines is different because this is a simulation. And even if we didn't have this stochastic event of extreme weather occurring, there would be stochasticity in the model runs given the stochastic nature of the models themselves. And that dark black line represents the average of those different colored lines. Slide 15. As I'd mentioned before, there are two main types of results that we draw from these models to explore how these stressors are affecting the population. And the second one had to do with the spatial distribution of individuals and spatial distribution of the population. And these are two snapshot images of a portion of the prairies of the South Puget Sound in Washington, where the Mazama park gopher is. The map on the left is year 2040 from a baseline scenario, and the map on the right, or the image on the right, shows gopher density from one of those extreme weather runs, one of those squiggly colored lines. The areas that are brighter blue on this map are where gopher densities are higher, and the areas that are red are where they're lower, and then the black areas don't have any gophers in them in this time period. So we can see that you can see how the distribution and densities of the population might change over time. Slide 51. So I'm now going to show you just a little bit of a few results for the tailored checker spot. And this is the model we've created for the checker spot. You can see the first thing you'll notice is this is a more complex diagram than the one I showed you for the Mazama pocket gopher. There are a few more things in that life cycle. It happens that the tailored checker spot butterfly has a more complex life cycle than the Mazama pocket gopher. And you can see that there are a number of factors that we're modeling that feed into that life cycle, some of which aren't necessarily stressors, but just have to do the biology of the species. One thing that's really important for all of the species on Joint Base Lewis-McChord is that habitat fragmentation and habitat loss for all the species I'm talking about, the habitat fragmentation and loss have been important, but for these three species, it's likely to be a key stressor. And so one of the things we're focusing on for the checker spot is movement, and we're focusing on restoration of habitat and reintroductions. And currently, this is a conservation reliant species, relies on regular reintroductions. And so we're exploring the degree to which the number of reintroductions, frequency of reintroductions is important, and how that interacts with restoration actions throughout the range of the species might affect the population. What I'm going to show you next in terms of results on slide 52 is habitat. We built a habitat model because habitat is so important for this species, and the fragmentation of that habitat is important. What you're looking at, there's a lot of Joint Base Lewis-McChord in that habitat map. And slide 53, please. We're also focused on movement, and another output we can get from these models is to look at how individuals are moving around. And the little black lines with arrows show individual movements. In this case, they're not dispersal movements, but they're movements of females while ovipositioning. And so in this case, there's multiple movement routines that occur in this model. Some of the models only have one movement routine, but we can have multiple movement routines that either for ovipositioning in this case, or for exploration of habitat by juveniles, or for dispersal movement. Slide 54. Here's some initial. So the two species I've showed you so far are species for which we have relatively little data compared to the amount of data we have for the three bird species in this study. So for the streaked-horned lark, the golden-cheeked warbler, and the black-capped vireo, we have a lot more data on the biology, better understanding of the biology of these species, but also demographic data and longer-term demographic data and better information about where the species is today. And so all that allows us to build models that are likely to be a little more realistic in the way they portray the actual population. It also allows us to investigate things in a different way. And so for the lark model, slide 55, please. We have data that's allowing us to model individual populations or parts of the population in a different way. So we have information about translocations and about population sizes at individual sites. We also have information about demographic rates at different sites. And so the model can be parameterized in such a way that each of these different sites has different demographics, and we can model the way birds are moved around between these sites. And so what you're looking at here it's a short-term projection, but it's just a 20-year projection, but it shows you the population size in these 9 different locations. And you can see that those are different. In this case, they differ mostly because emigration and immigration rates, not because of the population parameters, which don't differ in this model run. The other thing you'll notice, though, is that instead of just a single line showing the population, we've called out 4 different groups of individuals. And so we can track the whole population, which is what I've been showing you so far, or we can track different portions of the population, which help us gauge how well the model's working, whether they're establishing territories, who's moving when, and things like that. Slide 56, please. So I'm going to show you the most results for the golden-cheeked warbler, as we've been able to pretty much complete one set of scenarios, stressor scenarios, and run them through the model for this species. And there are a number of ways in which climate change is likely to affect the golden-cheeked warbler, but I'm going to show you one key way in which climate change is likely to affect vegetation, which in turn is going to affect habitat for the golden-cheeked warbler. And you can, slide 57. So the warbler relies on for habitat a mixture of ash juniper and deciduous trees, many of which are oak species. But the ash juniper, particularly mature ash juniper, is important for the warbler because it provides stringy bark that the warbler uses for nesting. So mature junipers are an important part of this bird's habitat. And there have been some models in the past that have projected climate impacts on the ash juniper, and particularly shifts in its distribution in space. And so one of the students at the University of Washington, graduate students, Leti Santillana, who's working on this project, has modeled the potential shift, climate-driven shift in the distribution of ash juniper. And she built a species distribution model, and then she's run a number of different climate projections through that to look at how this key element of habitat might change over time. The map on the left shows the projected current distribution of ash juniper, with the darker blue and gray areas being areas where we're more likely to find the tree. And then the yellow areas are where we're less likely. And what you see, the size of that map, the shape of that map, that's encompasses the current distribution of the golden-cheeked warbler. And if you look to the right, those 6 maps show projected future potential distributions within the range. And you can see each of those rows represents a different climate model, with the climate model at the top being the one that projects the least change, and the projection at the bottom, the one that projects the most climate change. The left-hand column of those maps shows the near future, and the right-hand map shows a projection towards the end of the century. And so you can see there's still quite there's likely to be quite a bit of ash juniper on the landscape in those top 2 maps, but when you look at the 2 maps on the bottom, that projection shows it largely disappearing from the current range of the golden-cheeked warbler. And I can show you that, then slide 58. This is a plot that basically shows that last slide in graphical form. So the current projection of the juniper, you can see at the bottom. And then as we look across models that are either project more climate change or project further into the future, you can see that the probability of finding the tree in the landscape decreases over time, or decreases with those projections. Slide 59, please. What Letty did was she took those projections of the change in ash juniper distribution and she used them to modify a current map of warbler habitat, saying, well, if the tree is likely to decrease in probability of occurrence across this map, she modified the ability of those areas to serve as habitat for the warbler. And then she did that at 5-year intervals and then fed those adapted habitat maps into the model and ran them for showing two of the different climate scenarios. Here they're from the same model, but they're from different emission scenarios. So the orange, there's an orange kind of squiggly line and there's a purple squiggly line. Each of those represent one model run, and then the curved lines or smooth lines. And you can see that the orange line, there's a bit of a turn up at the end where there's a bit less climate change projected under that scenario and potentially an increase in the distribution at the very end of that time period of the tree and an increase in habitat quality. The green line across the top represents a baseline scenario in which there's no projected change in the distribution of ash juniper. Slide 60. And then if we look at that spatially, specifically at Fort Cavazos, we can see the first thing that strikes you is that these maps look relatively similar for that large difference I just showed you. And the two maps on the right represent the orange line and the purple line. The map on the left is the green line. There is some difference on slide 61. You can see that if you look within those purple circles and then you look to the left at the baseline map, there is a decrease in occupancy, and you can see that in other parts of the map as well. But largely, the population doesn't change that much within the bounds of Fort Cavazos. And that's interesting because it does change throughout the rest of the range quite a bit. And so having a spatial depiction of how, in this case, climate change is likely to impact a key element of habitat is likely important because it lets us know that probably Fort Cavazos is likely to be important, very important for the management of the species in the face of climate change as the potential distribution of this key element of habitat changes. Slide 62. So with some conclusions, and this is one of the same conclusions that Enrico reached, climate change is likely to be a critical stressor. And we see that in the data I just showed you, the model runs I just showed you for the golden-cheeked warbler, but it's a stressor that interacts with most of the other stressors that we've got in these models. And it's going to be one that affects each of the species in these models in different ways. So it's going to be a complex stressor, and it's going to be likely to be a critical stressor. Second, for terrestrial species, loss of habitat and fragmented habitat are always important stressors, or almost always important stressors. And so fragmentation and loss of habitat and management strategies that can address those are likely to be the most important for the checker spot, the pocket gopher, and the lark. And then finally, we're finding as we get further into this that spatially explicit population models that can depict changes in not only the population size through time, but the distribution of the population through time are likely to be important to capture the effects of these stressors and multiple stressors at the same time. Slide 63. And in terms of some potential benefits to the Department of Defense, we're hoping that it provides an increased ability to anticipate stressor impacts and interactions between stressors. We're seeing this as an important tool for exploring management strategies. Not only can we look at different stressor scenarios, but we can look at different management scenarios as well. And we will provide a number of those as we complete these models, but the models can also be adapted to explore other management strategies. And then finally, these models will hopefully provide us with improved understanding of stressor interactions in general, how they interact, when those interactions are important, and when they're less important. Slide 64. And that's all I have. Thank you. I'm happy to answer questions.
Great, thank you so much. We have received a number of questions for you. The first one is from St. Mary's College of Maryland. When creating the scenarios you discussed, how much data do you compile and from how many sources? Is there a way you can condense it for easier modeling, or do you make some generalizations?
Yeah, that's a great question. And it kind of applies to the whole process, just not just the scenarios. So for some of the species and some of the scenarios, we have a lot more data than for others. So there's really a range of how much data we have for building any scenario or building each of these models. And so some of the scenarios we end up making larger assumptions. Some of the scenarios we're drawing on impacts from other species, congeners. Some of the scenarios we have very good data from our collaborator at Eco Studies who's worked on the lark for a long time, or from the biologists at Cavazos who've worked on the two species there for a long time. So sometimes we have to make generalizing assumptions, and other times we can have highly data-informed scenarios.
Wonderful, thank you. This next question is from the Lawrence Berkeley National Lab. Given that there is uncertainty in projections of future climate extremes, what is the most important information on specific climate extremes that climate researchers should focus on to inform their research?
Oh, that's a great question. And it's a great question because it's not the average temperatures and average precipitations, average annual precipitation that affects populations and individuals, but it's particular events and the accumulation of events. And so that's something that ecologists and biologists who work on climate impacts are wanting more and more, is good projections of extremes and extreme events. And it really depends on the species. Extreme heat events, drought is particularly important for a lot of species. So good projections of drought events, heat extreme events, heat domes and heat extreme events. Flooding's probably less, for the species I work on at least, flooding is probably not the most important of all of those, but any really freezing events, early freezing events can be important. I'm having trouble picking a few extreme events, but I'll just second the fact that yes, projections of those are uncertain, and that's something that's really important. And having projections of those is super important for understanding biological responses.
Thank you so much. This next question, if we can please go to slide 49. And we'd like to ask you, Josh, what weather events on the slide are represented? And can you say more about the relevance of the type of events?
Sure. Yeah. So this just generically shows any one of 3 extreme weather events. So for the Mazama pocket gopher, extreme heat events or extreme cold events while they're in their burrows can cause increased mortality, particularly of young, as can flooding with extreme precipitation events. Burrows can flood. We don't yet have a good idea of how much more frequent those events are likely to be. or exactly how the impact of each of those different kinds of events differs, right? And so which are more extreme. So this is one of the two species that we know less about, and so we're making more assumptions. But these data, this projection is sort of a generic increase in mortality while in the burrow based on a projected increase in any of these three types of events. So this is much more generic than maybe you're thinking it might be.
Great,
Hopefully
thank you.
Answers it.
A similar question to what we relayed to Enrico. Will the models you described be available for use and modification? And if yes, how easy are they to use?
Yeah. So the answer is yes. And these are, so the modeling platform to use these models is, so the models will be available, and the modeling platform is also freely available. It's something called HexSim. It was actually developed as part of a previous SERDP project. And so you can find HexSim on the web at hexsim.net, HEXSIM.net. And so I'd like to say it's easy to use, and it's relatively easy to use, but it's not super easy to build models. Even though there's a nice graphical user interface, there are lots of moving parts. So it's freely available, it's usable, the models will be too, one can modify them, one can download the software and build one's own models. Yeah.
Great, thank you. And then one last question that I would like both you and Enrico to talk about. Given what you've both talked about, how can DoD use information about how stressors interact to inform future actions? Let's start with you, Josh.
Sure. I think one of the key ways in understanding those interactions, if there are interactions among stressors where there's an outsized result, so there are synergistic impacts of stressors, one can use that information to target which stressors to focus management actions on, right? And so if there's a stressor that's combining with a lot of others and you're getting outsized impacts, maybe that's a stressor where you focus your efforts. Other types of interactions that maybe have compensatory results, maybe those aren't ones where one spends as much time and energy and money. So one way I'd suggest is that you could use it to prioritize management actions focused on specific stressors.
Thank you. Enrico, is there anything you'd like to add?
Well, I would like to second Josh's points. That's exactly what we've been arguing for as well. So this idea of coming up with combinations of stressors and evaluating different scenarios that also take into consideration the desire to keep an activity or a stressor while reducing others that are either feasible or more desirable to reduce. So I completely agree with Josh's point. And I think the models that we're developing will allow that. They will allow to test for those alternative scenarios that can be used to make management more effective. And it sounds like your group, Josh, are developing models that are, at least in principle, very similar to the ones we've been working on. So that's great to see.
Yeah.
Great, thank you both. And we received one more question for Josh that I'm going to relay to you, Josh, before we wrap up. This is from the Pennsylvania Department of Environmental Protection. Is your data solely based on projected climate changes, or are there other stressors included, like habitat loss due to development, pollution, or toxicity?
There will be, and from what I've showed you, most of what I've showed you are climate projection related stressors, but no, we'll have stressors that are related to training activities. We may have one toxin for one of these species, but we will have other stressors that have to do with habitat loss, habitat restoration, invasive species. Climate change is just going to be one of those. It'll interact with a lot of the other stressors, but we will indeed have other stressors that aren't based on climate change projection.
Great. Thank you both again for great presentations and robust Q&A sessions. We're going to go ahead and wrap up with a reminder that these webinars occur every 2 weeks, like Kevin mentioned, and the next webinar is on Thursday, October 10, on DoD-funded research efforts to study PFAS bioaccumulation in aquatic species. We'll have 3 speakers on that webinar: Dr. Celia Chen from Dartmouth College, Dr. Carrie McDonough from Carnegie Mellon University, and Dr. Jason Hoverman from Purdue University. Like Kevin mentioned, please visit the SERDP and ESTCP webinar webpage to register for this and other webinars through the end of 2024. Before we conclude, I would like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you'd like to refer to them in the future. We would appreciate it at this point if you can take a moment to complete a very short survey that will pop up on your screen. This concludes today's webcast. Thank you.